Hello guys, I am Shubham from Shubha Tutorials. This video is about ordered tetrad analysis. So before we get started, do watch the previous video to know the basics of tetrad analysis. What is tetrad? What is octad? Okay, and then continue with this video because in this video we are talking specifically about ordered tetrad analysis. So as we saw, the ordered tetrad is seen in Neurospora crassa. In this, there is a linear arrangement of spores or chromosomes we can see here in the picture. So basically, in this, there is different cases we will see. Suppose this is the case one, case one. Okay, in the case one, there is no crossover. Okay, no crossover. Then we will see this type of ratio. So 4 are capital A and 4 are small a. Okay, this is the ratio of no crossover, and this is known as FDS, first division segregation. Okay, FDS. So this is due to no crossing over. Okay, now if the crossing over occurs, then what we will see, that we'll see now. See, I will draw here again chromosome and pictures. Okay, this we are talking about case 2. Case 2, right? So this is a picture. Here there are two chromosomes. Okay, right? Okay, so these are the two chromosomes. We know that in the meiosis 1, there will be crossing over. So meiosis 1, right? After meiosis 1, there will be, these are AG, right? And these are small a. So there is a cross between these two. Okay, so we know that after crossing over, there is two cases seen. So one, right? So what will happen? There will be exchange of chromosomes. We know that basics of a recombination. This will be same. This will be same. And this will have some part of small. The exchange of chromosome, right? As well as there will be this exchange of chromosome. So these are now this is small a, capital A, small a. Okay, then capital A again, and then small a. Okay, so now after this there will be meiosis 2. Okay, after meiosis 2 there is a tetrad. We know tetrad. So here will be, okay, okay. So these are again, this is capital A, small a, capital A, small a, right? Then after the duplication, again mitosis, that is octad. Okay, this is a tetrad. I will just write down here then. First will be AA, then AA, A. Okay, so this will be octad, right? So this ratio is what? 2, 2, 2, 2. Okay, so do note that I have not drawn here. Okay, so I have just written here what will come after mitosis. Capital A 2, then small a 2, then again capital A 2, and then again small a 2. Okay, so this is the ratio in this case 2 of SDA. So this is known as SDA, second division segregation. Second division segregation. Okay, so this is case 2. Okay, this is in this SDS there is one more type. So in this case we saw the crosses between, I will compare it here. I will draw here again. So this is capital A and this is small a, right? We know the cross was between these two. Okay, in the case 2 of the SDS, SDS second division segregation, this is the first crossover. Okay, now the second type, in the second type, the crossover is between, crossover is between I will show you after drawing here. Again, I will draw the chromosomes. Okay, so now crosses between this and this one. See where I'm drawing carefully. Between first and fourth one. Okay, second and fourth. Here, second and third. Suppose I name here. These are the two chromosomes. Okay. Suppose this is 1, this is 2, this is 3, this is 4. So first was between these 2 and 3. Now, in the second case, it is between 2 and 4. 2 and 4. Right? So this is what we will see now. What will happen after the second division segregation. Okay. So this is second case of. Okay, so let's get started with the case second of the SDS. Okay, where there is crossover between second and fourth chromosome. Okay, chromatid we can say, right? So first of all, meiosis 1, right? Meiosis 1 will give rise to okay, there is exchange of chromosome, right? Then here again this is as it is, this will get changed, and these are packed inside a these are packed inside. Alright, after meiosis 1, then meiosis 2, meiosis 2. small a, this is again capital A, and this is small a, right? No, wait, wrong. This is small a, and this capital A here, right? So after this, what will happen is that there will be 4 1 2, I will draw till tetrad here, 3 4, okay. Then this is a whole chromosome, okay. This is a, okay. Then here there will be here, right? Right. So what is this arrangement now? This is capital A, this is small a, this is small a, and this is capital A, okay. After mitosis, now mitosis, so what is this now? This will form 2, after mitosis. So I will just write here a, small a, small a, small a, small a, capital A, capital A, right? So this is 2, then this is 4, and this is 2. So this is the ratio of second case of SDS, second division segregation. Got it? So this is what we want to understand of ordered tetrad analysis. So in the first case, I will just go through it. In the first case, we saw there is no crossover, that we will discuss in the basics, okay. So that method, there will be no crossover, nothing, just single meiosis, then mitosis, okay. Tetrad, octad will be formed, okay. So the ratio here is 4:4, okay. 4 capital, 4 small, right? 4:4 continuous in the linear. In the second case, where there is crossover, so FDS is the first division segregation where there is no crossover, right? FDS is the case 1 which we discussed previously, right? So here, so this FDS, no crossover, okay, that we discussed, okay. Then SDS, the second division segregation, have 2 types where there are crossover. First crossover is between the second and third chromosome, okay, and these are the ratios, 2:2:2:2:2, okay. In the second division segregation, case 1, this is the ratio, right? And the second case of SDS, there is crossover between second and fourth chromosome, and thus we see a ratio of 2:4:2, okay. Got it? Okay, so how to calculate map distance using this ordered tetrad analysis? So there is a formula, map distance is equal to half into number of SDS, okay, number of SDS upon total number of ASCII into 100. Got it? So this is the formula. Using this formula, we can find the map distance. That is distance between the centromere and the gene. Got it? So that's it for this video. I hope this video helps you. If this video helps you, do let us know in the comment section. In the next video, we will see about unordered tetrad analysis in detail. Thank you guys for watching this video. If you like it, please share it. Don't forget to subscribe and press the bell icon to get notified whenever the new video is out. Also follow us on all our social media pages, and you can join us on Telegram and stay connected for the latest updates. All links mentioned in the description section.